Hello students, this is my third video on the chapter cell for class 9 students. In the first video we had a discussion in general about the cell, its definition and the different types, shape, size, etc. In the second video we have discussed about the different parts of the cell and for some organelles. However, we did not do all the organelles, so this is my third video where we are discussing about the other organelles of the cell. So this was what we have been discussing from the previous video. We discussed about the boundary, cell wall and cell membrane, about the cytoplasm and the various organelles in which we discussed about the nucleus already. Now we are going to discuss about these other organelles which are present inside a cell. Mitochondria, one of the most important organelle, which is also called as the powerhouse of the cell. Now, do you know what is a powerhouse? Normally in electricity board we have something called as a powerhouse. It is that place where the power is generated or electricity is generated, there we call it as. Here we are talking about energy present inside the cell which is produced in this particular organelle. That's why we call it as powerhouse of the cell. So first point itself, we are saying that synthesis of energy in the form of ATP. Full form of ATP is adenosine triphosphate. Remember, ATP means adenosine triphosphate. It takes place inside the mitochondria by the process which is called as respiration. Now, if we look at the structure, this is a schematic diagram. You can see it has two boundaries actually. One is an outer boundary or outer membrane which is porous, but it is continuous. Continuous means if you see the inner boundary or inner membrane, it is folded, it is not continuous. So outer is continuous, inner is highly folded. Whereas outer is porous, inner one is selectively permeable. Now, inner membrane has certain structures. You can see here these dotted structures which are enlarged. It is a protein complex which is called as F0F1 protein. This F0F1 protein complex which is present in the inner surface is actually responsible for the generation of ATP process. The inner part of mitochondria is called as matrix. Whatever rest of the area inside that we have is called as matrix. It contains various enzymes etc. which are necessary for the various reactions required or carried out during the process of respiration. Here I would like to show you, this is an electron microscope structure. So this is what a real mitochondria look like. See, these are the infoldings that you can see, and these are certain molecules inside in the matrix. Now, function of mitochondria, we already said its primary and most important function is the synthesis of energy ATP molecules, and that is why we call it as powerhouse of the cell. It is a very, very important concept about mitochondria. We call it as semi-autonomous organelle. Please understand. Autonomous means what? Autonomous means a phenomena where the organism is kind of independent. It can do its activities by itself. It does not have to depend on something else. Now this mitochondria is called as semi-autonomous, means it is not entirely autonomous. Means it can do certain work by itself. For other work it has to depend on what? The most important organelle of the cell which we discussed in the previous video, and that was nucleus. So how it becomes semi-autonomous organelle is because it has its own DNA. Recall, where is the DNA present normally in a cell? It is in the nucleus. But mitochondria has its own DNA which is called as empty DNA or mitochondrial DNA, and its own ribosomes. Now what are these ribosomes? We'll come to it separately. So because of this DNA and ribosome that it has, it can produce certain proteins by itself which is necessary for respiratory reactions from its own DNA. But for other proteins, it has to depend upon the DNA which is present in the nucleus. So understand, for the respiratory reactions, there are a lot of different proteins which are necessary. Some proteins it can make by its own, so autonomous, and some protein it has to depend upon DNA, that is not autonomous, therefore it is semi-autonomous organelle. Coming to the next organelle, that is plastid. Remember, plastid is present only in plant cells. Now, plastids can be classified into three types, that is chromoplast, chloroplast and leucoplast. In some books you will also get that chloroplast is basically a specialized type of chromoplast itself. Now see, the word chromo means colored. So the plastids which are colored in nature are chromoplasts. They are colored because they have certain types of pigments. Leuco means colorless. So these plastids are colorless. Now chloro means green colored. Therefore, chloroplast is actually a kind of chromoplast, but it's a specialized type. So chromoplast is responsible for synthesis and storage of various pigments, and you find them in variously colored parts of the plant. Like you see brightly and differently colored flowers, different colors you find in it. Fruits you find different colors. Sometimes even roots and leaves also might be differently colored. So there you find these chromoplasts. Chloroplast, however, is the specialized chromoplast which is green in color because they have a special pigment known as chlorophyll. So these chlorophyll containing chromoplasts are actually called as chloroplasts, which are only and only found in the green parts of the plant and most predominantly in the leaves. Leucoplasts, like I told you, leuco means colorless. So they don't have any color, they don't contain any pigments. Their main function is storage. What do they store? Based upon what they store, they are classified as amyloplast, ileoplast and aleuroplast. Amyloplast is the one which is responsible for synthesis and storage of starch granules. Ileoplast is responsible for storage of fats, and aleuroplast is responsible for storage of proteins. Remember, amyloplast, ileoplast and aleuroplast responsible for amyloplast storage of starch, ileoplast storage of fats or oils, and aleuroplast storage of proteins. Coming to chloroplast, very important, it is that particular organelle where the process of making food by what is called as photosynthesis takes place in plants. Why? Because it contains a green colored pigment known as chlorophyll. Now they are found in all green parts of the plant and they are also double membranous like mitochondria. But here both the membrane, this is outer membrane, this is inner membrane. Darkly colored one is outer, lightly colored one is inner membrane. Both membranes are continuous. And inside matrix is called here as stroma, which contain various enzymes and other substances which are necessary for photosynthesis. Inside they have a specialized structure. These disc-like structures which you see are enclosed disc-like structure. They are called as thylakoid membranes. Now these thylakoid membranes can be present in stacks. Like you see, many of them, these are half cut, many of them are in stacked form. So one stack of thylakoid membrane is called as granum, whose plural is grana. Now inside this yellow colored, if you see, inside the lumen, which is a closed structure, flattened disc-like structure, it contains the pigment chlorophyll, which has the ability to absorb light and then carry out photosynthesis. So thylakoid membrane is therefore the site for the light reaction. Actually photosynthesis is divided into two parts. You will get it in next class 10. One part is called as light reaction, for which light is necessary. Other part is called as dark reaction, for which light is not necessary. Therefore, thylakoid membrane is the site for light reaction of photosynthesis, and stroma is the site for dark reaction of photosynthesis. So, this is again an electron micrograph structure of a chloroplast, and these are the stacks of thylakoid forming granum. Very much similar to mitochondria, again chloroplast is also called as a semi-autonomous organelle. Why? Because here also they have their own DNA. Here we call it as cpDNA or chloroplast DNA and the ribosomes, using which they can again synthesize some of the proteins necessary for photosynthesis and other proteins necessary for photosynthesis reaction. For that they depend upon the nucleus. Therefore they are again just like mitochondria, semi-autonomous organelles. That's a special feature of both mitochondria and chloroplast. Remember, it comes in the exam. This is one organelle which is not there in your book, but we keep discussing it again and again, called as ribosome. Now here it has two parts normally. This orange colored part is a large subunit of ribosome and this blue colored part is a smaller subunit of ribosome. Both together forms one complex of ribosome. They are not bound by any membrane, so they are non-membrane bound organelle. That is why they can be found in both prokaryotic cell as well as eukaryotic cell. They can remain either freely in the cytoplasm or they may remain attached to the endoplasmic reticulum, which are other tube-like structures we will discuss in the next topic. They are composed of proteins and ribonucleic acid. This ribonucleic acid is something which is very much similar to DNA, that is deoxyribonucleic acid. Now, since they are found in both pro and eukaryotic cell, they are of two types. In prokaryotic ribosomes, they are called as 70S type of ribosomes. Eukaryotic ribosomes are said to be 80S type of ribosomes. What is their function? Their main function is to carry out protein synthesis, that is produce protein, synthesize protein. So this chain which you can see is a protein which is being synthesized from a structure called as mRNA using a molecule called as tRNA. Now these molecules you are not studying in this class, this is a higher discussion. This is just to show you that the ribosome, these two units together, can carry out synthesis of protein, that is the main function. Coming to endoplasmic reticulum. See, these are some membranous tubular structure which are present in the cytoplasm, forming a kind of network. It spreads out through the cytoplasm. However, it is normally connected to the nucleus. Like you see here, this is the nucleus. From the membrane of the nucleus continues the rough endoplasmic reticulum and then smooth endoplasmic reticulum. They are of two types. Rough endoplasmic reticulum, they look rough under microscope. That's why you call it as rough endoplasmic reticulum. Why they look rough is because those ribosomes which we discussed before, they remain attached to their surface, these granular structures. Smooth endoplasmic reticulum, these ones, they look smooth under microscope, and that's why you call it a smooth endoplasmic reticulum. Why? Because they don't have any ribosomes attached to their surface. Coming to their function, before we go to the function, you can see once again the diagram. This is again an electron microscope picture, a part of the endoplasmic reticulum. You can see these tubes have these ribosomes attached, so they look rough in appearance. This is the nucleus, and these are some of the endoplasmic reticulum which are smooth in nature. Rough endoplasmic reticulum, most important function, they provide a surface for protein synthesis. Smooth endoplasmic reticulum, they provide a surface for lipid synthesis. Let me clarify here, many a time students do a mistake when we ask them what is the function of rough or smooth endoplasmic reticulum. They directly say rough endoplasmic reticulum carries out protein synthesis, smooth endoplasmic reticulum carries out lipid synthesis. Now this is wrong. Why? Because endoplasmic reticulum is not carrying out any synthesis. Synthesis is done by the ribosome, protein synthesis. But on the surface, Ribosomes are present on the surface of endoplasmic reticulum. So endoplasmic reticulum is providing a kind of site, a surface, a place where ribosomes can bind and carry out protein synthesis. Similarly, smooth endoplasmic reticulum provides a surface where certain specialized enzymes can carry out lipid synthesis. Smooth endoplasmic reticulum has another function in liver cells, specifically in mammals. The smooth endoplasmic reticulum is responsible for detoxification of drugs and poison. They remove their toxic effect. So, understand, this is very, very important. Protein and lipid we have studied in the previous video are two important constituents for membrane synthesis. That is, your cell membrane or plasma membrane or any other membrane in a cell is made up of protein and lipid. These are the two primary constituents. And these two things are produced by rough and smooth endoplasmic reticulum. Therefore, we say that rough and smooth endoplasmic reticulum is responsible for membrane biogenesis. Bio means living, genesis means formation. So it helps in formation of the membrane in the living world. That's why we use the term membrane biogenesis. So I am reading this out sentence. Since both SER and RER are responsible for helping in synthesis of lipids and proteins respectively, and lipids and proteins are constituents of any biomembrane, therefore ER helps in membrane biogenesis. Transportation of lipids and proteins. Now these are tube-like structures, so its one common function is to carry out transportation within the cell from one organelle to another organelle. Cytoplasmic framework, since it spreads out through the cytoplasm, so it acts like a framework for the cytoplasm where some biochemical reactions can take place on its surface. Next is Golgi apparatus, also called as Golgi bodies or Golgi complex. See, it's a beautiful diagram where we see that it has many flat disc-like structures which are stacked one above the other. Individual disc-like structures are known as cisternae, and these cisternae are stacked one above the other to form a complex. These cisternae are formed by fusion of these specialized vesicles. Now there are two phases in Golgi bodies. One is called as the cis phase, which is called as a forming phase, and trans phase, which is called as a maturing phase. Now Golgi bodies does not remain like that, it's not a static structure as such. On the cis phase, new phases keep getting added because of addition of these vesicles. See, it is written incoming transport vesicle. So some vesicles are coming from other part, getting added to it, and these are secretory vesicles, newly forming vesicles which are forming out. So this is the phase from which it is maturing out various vesicles. This is again an electron microscopic picture of a Golgi apparatus. Main function is modification of proteins and lipids. Please understand, we have studied previous endoplasmic reticulum. They help in synthesis of proteins and lipids, which carry out various reactions which make up various substances inside the cell. But when they are formed raw, these raw proteins and lipids cannot be functional. They need to undergo certain types of modifications. Now these modifications are taking place inside the Golgi apparatus. Some modifications such as proteins, they get associated with lipids and form called as lipoproteins. Glucose, they get associated with proteins, form what are called as glycoproteins. Glucose get associated with lipids, form what are called as glycolipids. Once these modifications are carried out, next function is packaging, because actually they need to do transportation of these things. Now when you send something to some place through a courier, do you send it naked? No. What do? You package it. You put it inside a box. So the packaging takes place inside those vesicles, and then those packed vesicles are transported either to a different site inside the cell, or it may be shown outside the cell. Another special function of Golgi apparatus is to form one more type of special organelle called as lysosome. So let us look at lysosome. Lysosome is also nicknamed as suicide bag of the cell. First of all, what is it? It is a membrane-bound spherical organelle. The membrane of the lysosome is this one, which is single wall membrane, not a double wall membrane, and they contain various enzymes inside which are called as hydrolytic enzymes. They have an ability to digest carbohydrates or proteins or fats as such. So what is their function? Their function is to engulf and break down any worn out organelles of the cell. Organelles, if they are no more functioning, they are worn out, they need not be present inside the cell, they are useless, so they will be engulfed by it and then it will be digested. Secondly, they can also break down any pathogenic organisms, any disease causing organisms that might enter the cell, or any harmful food material also, if it enters something that is not necessary that may enter the cell, can be broken down by engulfing. Now, since its function is to engulf harmful substances or useless substances inside the cell, it has a special function. What is it? If the cell is damaged, is injured, suppose, and it's no more functioning, then it is not required. It needs to undergo death. Then what happens is this lysosome will burst out. When it bursts out, the inside hydrolytic enzymes are released out. When it is released out, then those enzymes will digest all the other organelles present in the cell. And therefore, when organelles are dead, what will happen? The cell will undergo death. Cell will no more be functioning, so it is dead. It is dissolved. So that's why we call it a suicide bag of the cell, because it is responsible for killing its own cell, not the normal ones, the ones which are damaged beyond repair. Next organelle is vacuole, the last organelle that we are discussing. Vacuole is a membrane-bound spherical organelle whose main function is storage. Large vacuoles you can find inside plant cells. See, this is the plant cell. Inside you can see a very large vacuole. Usually vacuole can take up huge area inside the cell. 50 to 90% of the cell volume may be taken up by one single vacuole, thereby pushing all the other cell organelles towards the periphery. In animal cells also they may be present, but usually small in size. When we look at function, in unicellular organisms, there are some vacuoles formed called as food vacuole. If you recall, pseudopodia, they engulf food within a food vacuole. So they are used for engulfing the food and its digestion. Unicellular organisms like amoeba and others also have a specialized vacuole called as contractile vacuole. They use this contractile vacuole for throwing out excess water which might be present inside the cell. In plant cell, they normally contain cell sap. What is cell sap? It's water plus dissolved minerals, etc. And they are responsible for maintaining turgidity and rigidity of the cell. Turgidity means it keeps the cell membrane fully extended. They also store substances such as amino acids, proteins, organic acids, sugars, etc. That function they can also carry out in animal cells. Now, from last year we have another topic added to this particular chapter, very important topic called as cell division. But we have only a brief understanding of this particular topic. Now, cell division, as the name suggests, it is nothing but the cell divide or break down or multiply. Now, for the formation of new cell, recall cell theory, where Rudolf Virchow said that new cells can arise only from pre-existing cells. So, how will a new cell appear from pre-existing cell? That pre-existing cell need to undergo division and give you new cells. So there are two types of cell division we observe. One is called as mitosis, other is called as meiosis. Now, mitosis is a cell division where a single round of division takes place. Meiosis, however, in each division there are 2 rounds that takes place. Single round means mother cell will divide only one. Mother cell is the originating cell. That will divide only once and give 2 cells which are called as daughter cells. In meiosis, that mother cell will divide first once, give 2 daughter cells. They again divide to give finally 4 daughter cells. So, these 2 daughter cells in mitosis, they will have equal number of chromosomes, and that is why this type of a division is also called as a equational division. Remember this word. If you remember this word, you can easily understand this particular difference, which is very important. Equational division means the daughter cells will have equal number of chromosomes as mother cells. See this diagram. Here, these individual chromosomes, these are 4 chromosomes which are present. This blue color is 1 chromosome, this is another chromosome, this is 1, this is 1. So 4 chromosomes. Each chromosome is present as a doublet. It is duplicated chromosome. But here, daughter cells will again have 4 chromosomes as singlet. So out of these 2, 1 goes to 1 cell. That's why 4 chromosomes remain as 4 chromosomes in the daughter cells. That is equational division. In meiosis, what happens here is, here also you see 4 cells. First it divides into 4 chromosomes, dividing into 2 chromosome, but from this one again each chromosome is dividing into 1. Therefore, you finally get 4 cells having 1 chromosome each. That is why we call it as reductional division. Here you have 4 chromosomes in the mother cell. Here you have only 2 chromosomes per cell. So it is reduced to half. Now, this is the process by which special cells necessary for reproduction called as gametes like sperms, eggs, they are formed by the process of meiosis. But for general growth and repair, when you are growing taller and taller, newer and newer cells are forming, that is growth. When you get some injury, some skin is removed. So after a few days you will see new skin is formed. So that new skin is formed by a process called as repair, where the new cells are formed. So for this growth and repair, it is the mitosis or mitotic division that takes place. So this is the difference between 2 types of cell division known as mitosis and meiosis. Coming to the hints for the in-text questions. Can you name 2 organelles we have studied that contain their own genetic material? It's mitochondria and chloroplast. Why are lysosomes known as suicide bags? Because they can destroy their own damaged cells. Where are proteins synthesized inside the cell? On the surface, not inside, but on the surface of rough endoplasmic reticulum. What would happen to the life of a cell if there was no Golgi apparatus? So think about its function. We call it function. Its function was mainly to modify the proteins and lipids. So those modifications will not happen. They will remain non-functional proteins and lipids. Which organelle is known as the powerhouse of the cell? Mitochondria. Why? Because they can generate ATP, that is the energy currency. Where do the lipids and proteins constituting the cell membrane get synthesized? Again the same question, endoplasmic reticulum surface. Which type of cell division is required for growth and repair of body? Which type is involved in the formation of gametes? That is for the growth and repair mitosis, for gamete formation meiosis we discussed just now. Some extra questions that you can think of at this level is what is the function of vacuoles, or for that matter you need to study the function of all the organelles. What is a contractile vacuole? What is the meaning of membrane biogenesis? Very important question. What are the toxics? Where are the toxic substances detoxified inside a liver cell? What is a ribosome and what is its function? This is not in your textbook but very important, you must know about ribosome. Why is the inner wall of mitochondria highly folded? Name the plastids responsible for storage of carbohydrates, fats and proteins, all those type of leucoplasts. So students, that was all about the first chapter of biology in class 9, that is the fundamental unit of life, cell. We have discussed it in 3 videos. In my next video, we will be discussing about the second chapter of biology, that is tissues. Tissues are formed by aggregation of the different cells. Now, for the next video, you kindly subscribe to my channel, like my channel, share it among your friends so that more students get benefited. And again, if you have any doubts for clearing them, feel free to contact me over my WhatsApp number 94353 52781. Thank you so much, students. See you in the next video again. Thank you.